Now, so far, all of our examples have really been about genes that have only two alleles. And it's really easy in that case because you can have either the dominant recessive, the codominant, or you could have that incomplete dominance. But not all genes have only two alleles. Some genes have more than two, so they could have three or more. And so that's when you have three or more alleles of the same gene, the way that the inheritance works is a little bit different. And in this case, you can't really look at it on an individual basis. You have to really consider the entire population to get an idea of how the inheritance is working. And the classic example of a locus with multiple alleles is the human ABO blood groups. So in the case of the ABO blood groups, you have additional antigens on the surface of your blood cells, sort of like the M and N. Right. There are three different alleles. One of them is a loss of function, and that's type O. So type O, if this is a type O blood cell, will not express any antigens on the surface. Now the other two, A and B, actually show a different type of inheritance pattern. And so I was gonna draw it here, but I'll work on it on the next slide instead. So the alleles are called IA, IB, and little I. So little I is the loss of function. So A and B make their respective antigens. So the IA gene makes the A antigen, the IB allele makes the B antigen. The little I is a loss of function. Now in terms of relationships, both B and A are dominant to the O, the lack of an antigen. So if you have an individual who has IA, little I, if this is their genotype, their blood will look like this, A. That's great. And if you have an individual who has IB, right, with the lowercase I, their blood cells will be B, right, B. And then if you have this poor person here, like me, their blood cells they don't have anything. Okay, so that illustrates this relationship right here, that the A allele and the B allele are both dominant to the O allele. Okay, cool. So then what is the relationship between the A and the B? What happens if somebody has A and B? So A is dominant to O, B is dominant to O. How do A and B interact? They are co-dominant. And what that means is that somebody who has AB blood will, just like with our MN, they will put A and B on the surface. So we have three different alleles, and we can get four different phenotypes: A, B, O, and AB. So we have complex genetic relationships. We have a classic dominant recessive if we compare A and O or B and O, but then we have codominance when we compare A and B to one another. Now, again, you have the four possible phenotypes. You have 6 possible genotypes. So you can be homozygous for A, heterozygous for A and O, homozygous for B, heterozygous for B and O, heterozygous for A and B, or homozygous for O. And so these two, as long as you've got two A's or an A and an O, your blood type will be type O. If you have two B's or a B and an O, your blood type is B. If you have AB, you have AB. And if you don't have anything, you don't have anything. Now they represent carbohydrate groups on the surface of red blood cells. And again, if you have type O blood, you don't have those special groups. You only have kind of the standard thing that shows up on cells. Okay. Now there are also some weird exceptions. So the reason that we don't like to do blood typing in class is because sometimes blood typing can identify relationships that are incompatible with science. For example, two parents who have type O blood. Let me draw that out here. If two parents have type O blood, their children should all have type O blood. If their children have type A or B, someone or both are not the parents. Okay, so that's, or very likely not the parents. That's when it gets a little bit dicey, and we don't like to do this in class. Okay. Now there are some genetic conditions where you could have a offspring. This is an example of a classic family. They call it the Bombay phenotype. The female phenotypically, right, has type O blood. So we think genetically she should look like this. Well, why is that a problem? Well, we're going to see the problem on the next slide. So what we see on this slide is that we have a type A parent and a type AB parent. But the children that result are AB, O, A. Oh, sorry, this is not one of their children. That he married into the family. Excuse me. Okay, so we see AB, O, and we see type B blood. Okay, so why am I saying that this is weird? Well, this is mom, so she can give A or B, right? IA, IB. This is dad, for sure is going to give an A. We don't know what's going to be over here, right? That's fine. Okay, so now if we do our Punnett square, even if we don't know the other allele that dad has, that's okay. So this individual, right, would look like this. This individual would be this. This individual would at least be this. This individual would at least be this. So the idea is, if you have a parent who has type AB blood, genetically, you cannot have a child with type O blood. Because there is, to get type O blood, both parents have to at least be carriers, and an AB parent is not a carrier. Now, okay, well maybe this isn't really the mother, right? Okay, let's assume that. Okay, so now we look at this individual here who married somebody with type A blood. So let's do this. So she looks like she should be this, right? She should be recessive for the blood type. And the father is going to be IA, maybe with a question mark, right? Because we don't know yet what that other allele is. So if this woman is truly II, she's got O-type blood, she marries a man with A-type blood, and they have children. Okay. We should get some that have type A. These will be carriers for the recessive, right? but we don't know what else they could be. So let's look at her children. Well, I'm looking at her children and I'm seeing the B allele present in 2 of her children. So then, okay, the father must have it, right? Because she's type O, so he must, wait, he's only type A. That means he can't have the B allele. The B allele had to come from the mother. So genetically, she can't be this. She can't be genetically type O. She has to at least carry 1 copy of the B allele. But why does she look like she's type O? Why are we seeing this? Well, the answer is on the previous slide. When they looked more deeply at this woman's genetics, they found that she had a mutation at a gene called fucosyltransferase, FUT1. Now, what happens here is that when you make your blood, you put these little things called H substances on the surface, and then that's where the A and the B attach. It's like the little hook that allows A and B to attach to the cell. Now, this person, when you have a mutation in this gene, she actually can't put the H on the surface of her cells, so it's not there. No H, there's nowhere for the A or the B to attach, so they can't bind either. So even though genetically she should at least have some type of type B, phenotypically she looks like type O. And so why do I go through this big example with you when it's just, this lady that has this weird kind of quirk? Because I really want you to understand that, one, just because something looks a certain way doesn't necessarily mean that the genetics reflect it. But 2, you can have multiple genes that interact. So you have the gene that encodes for the antigen, and you have the gene for this sugar transferase that both are responsible for developing the blood type. When this gene is not functional, the person will look type O no matter what their blood type genes say. So you can have multiple genes influencing a single phenotype. And so genetics are a lot more complicated than we would like to think. Now we can also have genes that we call lethal alleles. And so what happens with a lethal allele is that the gene product, whatever that gene makes, is absolutely required for survival. Generally, what happens is that an individual can tolerate being heterozygous. So if the mutation only affects 1 copy of the gene, only affects 1 allele, the individual should be okay with a single wild type allele. However, an individual that inherits both of the recessive alleles will not survive. And so whatever mutation happened to that allele makes the allele be a recessive lethal allele. And these are relatively common, right? There are lots of examples of recessive lethal alleles. But for the most part, the actual lethality isn't common because you'd have to get 2 individuals together that are heterozygous, that are carriers for it, and then they would have to have an offspring that had that 1/4 chance of inheriting the homozygous recessive. So they're not as common as they could be, I guess. But you can also have situations where the mutation causes a lethal allele to be dominant. So anything lethal can kill the organism. Now, in a case like Huntington disease, this is considered to be a dominant lethal. And what that means is that if an individual, right, has the dominant form of the allele and the recessive, this individual is going to die. This individual is also going to die, probably as an embryo. This individual is the only one who lives. But when does this person die? Unfortunately, this person usually dies in their 40s or 50s. So after they've reproduced and likely passed on this allele to the next generation. So you only need one copy of it for it to be lethal. So it's a dominant autosomal allele. In heterozygotes, the disease is delayed until adulthood, and the symptoms are progressive neurodegeneration. So it's a neurodegenerative condition that leads to things like dementia and, of course, an early death. And again, that's inherited in a dominant form. There's another allele. This is in mice. It controls their coat color. A gouty, I don't know why they call it that. It's brown. To me, it's brown. It's like a brown mouse. But there's also a mutant yellow allele. So in terms of its behavior, the mutant allele is actually dominant. And we're going to see this on the next slide, so don't worry about it. However, we say it behaves as a homozygous recessive allele. And the reason we say that is because if you are, or if you're a mouse and you're homozygous for this yellow allele, you die. And so what we're going to see on the next slide is kind of more about how that works. So you have, see, it's brown. They call it a gouty, but that's fine, it's brown. So when you have two agouti mice, you know that they have to be just plain A because the yellow is dominant. And so you have the two agouti mice, you cross them, all the offspring are agouti, they all survive. Look at this cute little brown mouse. If you cross two heterozygotes, they're both going to be yellow because the yellow allele is dominant. You cross two heterozygotes, you get the what you would think would be the 3-to-1 ratio, right? You should have 3 yellow to 1 agouti, but what you actually have happen is you end up with what we call a 2-to-1 ratio. And you get this 2-to-1 ratio because the homozygous yellow dies. And so you have a 2-to-1 ratio. So anytime you have an allele that 2 copies of it kill the organism, especially kill it like embryonic lethal, you get a 2-to-1 ratio. So if the embryo wouldn't survive, you see this 2-to-1 ratio rather than a 3-to-1 ratio. If you cross the recessive with the heterozygote, you get half that are yellow, half that are agouti, and they all survive. Okay, so here's what your yellow mouse looks like. So when you are starting to look at genes that are lethal, not just recessive, they could be dominant, but when you put 2 of them together, they're lethal, you get different ratios, especially when they're embryonic lethal.